This method can help with investigation of membrane proteins, such as transporters and channels, and the information about substrate turnover rate allows comparing protein activity and specificity under different physiological and pathological conditions.
The main advantage of this technique is that it minimizes potential shift variation in chloride-free buffer conditions and significantly increases precision of turnover rate determination due to more exact membrane potential measurements.
Gather the elements for the first steps. These include 2 microcapillary pipette tips, a sharp blade, and a sliding caliper. For the electrode, have silver wire, sandpaper, and a 3 molar potassium chloride solution at hand. In addition, have ethanol and water for cleaning and a DC power supply. Proceed by working with the 2 micropipette tips. Start with the tip that will contain the buffer. Place the second tip next to the first. Move the second tip so, when inserted, its narrow part will extend at least 5 millimeters into the narrow part of the first. Mark the position where the first buffer-containing tip will be attached. Then, use the caliper to measure the length of the Micro-agar salt bridge electrode. Use the blade to cut the tip to the appropriate length. Clean the cut surface with ethanol followed by water. Next, get a length of approximately 8 centimeters of silver wire for the electrode. Clean the wire on wipes with ethanol followed by water. After cleaning, use sandpaper to smooth the